I think the title sort of gives this away, but I'm hoping you can understand. I'm going to talk about an organ that most of us have in this room. So this is the brain. So the brain takes up roughly about 2% of our body weight, but actually uses more like 20% of the energy that we require. And to accommodate that, we have this very complex neurovascular unit which sort of facilitates the blood flow and facilitates the oxygen, the nutrients that we need. And of course, normally flying around this beautiful vasculature, we have heaps of immune cells until, we get a clot. And all of a sudden, we get the inhibition of the perfusion of this neurovasculature. And then obviously we know this is now going to cause a very large ischemic region in the brain. You can see this sort of black core that we get, and we're under no illusion this black ischemic core is probably not going to be salvageable. We can't bring dead tissue back from the death. But what we can try and salvage is this ring around it, this inflammatory region which we call the penumbra. And to try and treat this, as we know, I'm going to bring up this diagram again as everyone else is. We've got TPA, which can try and enzymatically disrupt the clot, and we also have endovascular clot retrieval, and where we try and pull out the clot. But as we know with both of these methods, we have the very hard challenge of time. And despite the hard work, which we saw this morning from Bruce or from Henry Marr over at Monash Health, where we're trying to increase the time and the eligibility of patients, we always get these issues where we can't really salvage these patients and get them back to full health. So rather than targeting the clot, which a lot of people here are obviously doing, we're trying to take a slightly different spin on this, and we're trying to target neuroinflammation. So we're trying to save the penumbra. And this is this region here between the ischemic core and the inflammatory ring which surrounds the ischemic core as well. And again, as we've seen multiple times today, I do photothrombosis, and we try and target this sensory motor cortex, which sits just here, so just off from the bregma point here. And to do this, we inject the photosensitive dye, rose bengal, and then we excite this with a cold laser, which is a green laser. And we can then actually watch these clots form. So as Connie showed this morning, we're very fortunate we have intravital imaging at our disposal. And in the cyan here, we have the platelets, and they're white at the moment because they're mixed in with the rose bengal. And we can see the rose bengal slowly disseminates, but it's activating the endothelium. We're getting platelet recruitment, and we get localized thrombosis. And we can determine exactly where this ischemic region is actually going to be. And we can see at this point we have full occlusion of the vasculature, and now we have a stroked mouse. And 3 hours after a stroke, when we do a TTC staining, which stains for the live region of the brain, we can see the local core is actually quite small in that top right corner there. And 24 hours later, we get develops to like a 30 sort of millimeters cubed region. And when we quantify this across multiple mice, we see this sort of therapeutic window between 3 and 24 hours, where at 3 hours we get that unsalvageable core, but it hasn't quite reached that really large 24-hour region at this point. So can we stop this from happening? And first we wanted to look at the vascular integrity after a stroke. And we did this at multiple time points by injecting Evans blue at intervals after a stroke. And we tried to see how much Evans blue had disseminated into tissue. And when we did this, obviously in the sham mouse, we get a perfect blood-brain barrier. There's no dissemination of the Evans blue. But within half an hour of a stroke, we can see this Evans blue is already leaking out of our vasculature and into the tissue. And then by 3 hours, we can see this sort of red region, which sort of identifies our ischemic core. And surrounding that, we still get that leaky vasculature. And by 24 hours, this vasculature is completely occluded. And at this point, it's really not very salvageable. And we can quantify this across the multiple time points. And we can see that sort of 3 to 6 hour time point is when the vasculature hasn't occluded yet, and it's still very, very leaky. So what else do we know about this? So if we've got leaky vasculature, what does that mean for the state of the brain? And we had some help, and we did some MRI imaging of the brain. And this is the very front of the mouse, and I'm going to flick through the different regions here. And we can see the edema in the mouse popped up there, and I'll take a screenshot of that in the ipsilateral side of the brain. So we can see in the top right is lots of water, so it's very low dense, so it shines up beautifully by an MRI. And we can see on the left-hand side is the contralateral side, and you see there's no water there at all. And what are the implications of this edema? And we tried to assess that by doing some motor function testing. So we suspend mice from a very small wire, which is roughly 40 centimeters off the ground, so it's high enough they think they have to hold on. And we can see how long can they hold on to this wire for. And we show that a healthy mouse could hold on to this wire for roughly about 90 seconds. However, after a stroke, within just 3 hours, they fall off within 20 to 30 seconds. So it's a really, really impactful and strong phenotype which we're seeing. And that then continues over the next 24 hours. We don't really see much worsening than that. So can we sort of stop this from happening? Can we stop this edema? Can we stop this inflammatory event? So we wanted to identify what are the peripheral immune cells in the brain. And so we did this by developing a 32-color spectral cytometry panel. And we looked at heaps of immune cells. We tried to characterize them and work out what's going on in our brain. So the most obvious thing to look for here is going to be the neutrophils, the monocytes, and the monocyte-derived macrophages. And we showed that after 3 hours, on the left-hand side we've got the sham, on the right-hand side the stroke, and within those compartments we have the contralateral, which remember is the healthy, and the ipsilateral, which is the stroke-induced area. So we have a very small infiltration of neutrophils within 3 hours of a stroke. We have a small infiltration of monocytes, and a small population of monocyte-derived macrophages. So we're not really seeing much immune cell infiltration into the brain after a stroke. But after 24 hours, this is massively exaggerated. So we see heaps of neutrophils, monocytes, and monocyte-derived macrophages. So can we assume this is when we're seeing the most inflammation? But I think from data we know that's not the case. We see inflammation way earlier than that. And when we quantified the cytokines in the brain at different time points, we looked at the cytokine composition at 3 hours and 24 hours in the ipsilateral brain. And we saw what was quite alarming. After just 3 hours, we're seeing heaps of IL-1 alpha, beta, IL-6, and TNF alpha. And this is a really rapid response. So is this why we're seeing this really harsh motor neuron deficit within just 3 hours? So we thought, okay, if these are the problem, let's get rid of them. So we then started to deplete myeloid cells, and we did this using a strategy of anti-GL1, so it binds onto Ly6C and Ly6G, and we're then going to attempt to remove all myeloid cells from the periphery, from circulation. And we can see in our IgG control, we can see by a classical CD45 against 11B FACS plot, we can see heaps of myeloid cells circled, or squared, there in blue. But in our anti-GL1 group, we see that they're almost all eliminated, and we actually see a 99.9% depletion of neutrophils and a 79.8% depletion in monocytes. So have we saved stroke? And of course that's not going to be the answer, because when we look at those nice cytokines, when we look at IL-1 alpha, IL-1 beta, IL-6 and TNF alpha, in the IgG control they're still there, and they remain in the GR1 group as well. So we haven't prevented that inflammation. And similarly, when we do the hanging wire test again, we see no improvement of these mice in hanging onto the wire. But we do accept the depletion of monocytes, it's not perfect. And when we look at that population, if I was to remind you, we see loads of monocyte-derived macrophages in the brain after a stroke. So using this huge multicolour panel that we've got, we looked at this population and we saw they had really high expression in CD80, 86, MHC2 and INOS, those really nasty markers of an inflammatory macrophage. And we saw a downregulation in CD206, ERG1 and EGR2. So we know these are a very aggressive type of monocyte-derived macrophage. So we thought, okay, let's get rid of these. So we used the CCR2 knockout mouse. So these monocytes now can't respond to classical inflammatory stimuli. They can't extravasate in response to a stroke. So we looked at our brains. So here's our wild-type brains. And as always, we see the infiltration of monocytes and monocyte macrophages in response to a stroke. But in our CCR2 knockout brains, we don't see any infiltration of monocytes and monocyte-derived macrophages. So, again, have we cured stroke? But alas, no. So we still see heaps of IL-1 alpha, IL-1 beta, TNF, and IL-6. So, and alongside that, we see no improvement in the hanging wire score again as well. So if we get rid of our peripheral immune cells, we don't see any improvement. So let's just recap on that. So we know that immune infiltration occurs more than 3 hours after a stroke. We know that we see a pro-inflammatory cytokine profile less than 3 hours after a stroke. We know that peripheral immune cells don't contribute to the development of acute neuroinflammation. So what could be driving this super acute neuroinflammatory phenotype that we're getting? So let me introduce a cell type which we haven't really heard much about today. So I'm going to talk about microglia here. These are the brain-resident macrophages. And these are my favourite cells. They're beautiful. They're huge. They're dendritic. They're very good at surveilling the brain, so they can prune neurons. They look after them. They remove cellular debris throughout all stages of life. And they look like this. They spin as well, apparently. We're very fortunate. We have the CX3CR1 GFP reporter mouse, so we can watch these microglia live by imaging. And we can see as well, we can discriminate spatially in response to a stroke. So we can take what these cells look like at different regions of the brain after a stroke. So you can go to the far left, to the contralateral tissue, and we can see that these are the healthy region, and we can see what they look like here. And then we can get so slowly closer and closer to the infarct in the top right to see how these cells morphologically look in response to a stroke. And when we do this, we can see that in the contralateral tissue, as we might expect, they're still beautiful. They're huge, they're dendritic, and they have these nice complex legs which come off from their cell body. But When we go right into the infarct core, we see they're way less complex. So they have way less dendrites, way less levels of dendrites, and they also have an enlarged cell body as well. And we can quantify this spatially across the brain, and you can see the closer we get to the core, so left to right, we have less cells, we have a larger cell body, and we also have less complexity, so each cell has less dendrites. But we very much appreciate that we can't define what a cell does from its morphology, so we're trying to delve deeper and deeper into this. And as Connie showed earlier, she actually stole my video here, so it's a little bit less impactful, but if we can watch these strokes happen live, and we can see the laminar flow, or the turbulent flow, I should say, of blood trying to get down this ischemic vessel, and we can see the platelets there sort of giving it a go, but then turning back and going a different way. And we can also do this in conjunction with microglia. So we can see here the microglia in green, and we can see the platelets in cyan, and we can sort of be like Luke Skywalker here going through the trench run of the Death Star, and we can sort of delve in and out of these blood vessels, and we can see the microglia are slowly trying to interact with the vasculature. We know that they're trying to migrate towards it, but they're also a little bit unhappy, so they're morphologically dissimilar from the contralateral side of the brain. So we know this is a very, very unhappy region. So is this targetable? And I sort of alluded to this earlier when I spoke about vascular integrity, but I sort of brushed over it. But we know in the brain we have the blood-brain barrier. And we know this is made up of endothelial cells, neurons, astrocytes, perivascular macrophages, pericytes, and of course, microglia. So we wanted to assess, we know all of these cells are already in the brain, so what does each of these cells do in response to a stroke? And we know we already see this phenotype with microglia. So we started to harvest the brains, and then we cultured them for 3 hours of all of these cells in the presence of Brefeldin A. So we tried to work out which of these cells is responsible for causing these cytokines being secreted in the brain at such an acute time point. So this Brefeldin A will keep the cytokines in the cell, and then we can quantify it by flow cytometry. And we looked at astrocytes, pericytes, endothelial cells, and we didn't really see much pro-inflammatory cytokine secretion from these cells. However, when we looked at microglia, which we'd cultured with Brefeldin A, we saw an increase in the secretion of IL-1 beta, in IL-6, and TNF-alpha. And keep in mind, this is after cell culture, so all of the super unhappy microglia will probably die in the process of brain sectioning, and brain culturing, and brain processing. So these are the cells which survived, possibly the more happy cells which are going to be in the brain, and they're still secreting this much of the pro-inflammatory cytokines. So if we just wrap this up real quick. So we know that pro-inflammatory cytokines are detected in the brain before the arrival of the peripheral immune cells after stroke. We know that microglia morphologically respond to stroke within just 3 hours. And we know that microglia are fundamental in driving acute neuroinflammation in response to ischemic stroke. So perhaps a strategy targeting microglia in response to stroke might be the way forward in salvaging that penumbra region. And with that, I'd like to thank all of our collaborators, especially the Wong Lab and the core facilities, and a special thanks to the funders, including the CAS Foundation, Heart Foundation Vanguard Grant, and AVBS. Thank you very much for your time.